Welcome to NewYorkBIO's virtual breakfast series, a digital program started in 2020, bringing you fireside chats with leaders from across the healthcare spectrum. This week's episode features William Ho, the co-founder, president, and CEO of IN8Bio.
All right, I have 901. Good morning, everybody. My name is Jennifer Hawks Bland, and I am the CEO at NewYorkBIO. We're thrilled you're here to join us in our conversation with Will Ho from IN8Bio. We plan to cover lots of topics today, and we wouldn't be able to cover any of these topics without the support of our sponsors. So thank you, especially to VWR, who has been our longtime business partner in helping to provide discounts on lab supplies to all of our member companies. So we really appreciate that relationship and what they are able to do for our community. So thanks to VWR. And with that, I will remind everyone during our conversation with Will, please ask your questions either in the chat or the Q&A box. And that way we can get to them as we can and put them in at the appropriate place. And it wouldn't be a virtual breakfast series leading up to our annual meeting without me mentioning that our annual meeting is coming up on September 27th. We will be hybrid, and as I had mentioned before, we made the decision, irrespective of sort of what the state or local requirements are, that if you plan to attend in person, you need to be vaccinated and be able to show proof of vaccination against COVID-19. So we look forward to welcoming many of you in person, but for those that are not able to join, we will have a virtual option so that you can still join us and participate. With that, I will turn it over to Derek to do a better introduction of Will, and then we'll have our conversation. Thanks, everybody.
All right, thanks, Jennifer. And Will, good morning. It's great to have you here. I love the idea of company creation stories. I love New York City company creation stories. And I've followed this one for a long time. As a fun fact, I think it's about 12 years ago now, I had started this networking group for biomedical entrepreneurs. And I believe you were one of the people on, the original invite list and the original first ever meeting of that group. So it's always good to wind back that clock. So thank you so much for joining us. We're super happy to have you today.
Thanks for having me. I appreciate it. Both you,
Yeah.
Derek, and Jennifer.
So I want to get into all things IN8Bio, but kind of the first thing that we like to do is wind back the clock a little bit, have a little bit of an origin story from our guests. You've got a great background. You started in finance. You were at New Leaf for a number of years, so you basically made this transition from kind of pure finance all the way over onto the entrepreneurial side of the equation. So can you talk a little bit about how you got there, because there's really no standard path for how anybody gets where they are.
Sure. I guess, truth be told, I had never really had an intention of becoming an entrepreneur or a biotech CEO. It was completely somewhat by accident. I'm a biochemist by training. After business school, I actually started my career in healthcare investment banking at what's today known as Cowen. At this point, I've been in biotech for about 20 years. I'm pretty unique in that I've been in pretty much most aspects across the business. And so I went from Cowen after 9/11, ended up at one of the early genomics company, Curigen Corporation, running operation analysis and FP&A. Unfortunately, as you remember, there was a big biotech boom, and then it popped.
Yes.
At Curigen, I was responsible for initiating their two first RIFs or reduction in force. I see some of my old colleagues are on this call. It was a tough time, tough decisions, but we had unfortunately reduced the staff by almost 250 people.
Oh my gosh.
After the second one, I made the decision to go back to Wall Street. I started off in equity research. I was at Piper, was eventually Bank of America's lead biotech analyst, until the financial crisis. Right after the financial crisis,
like that.
The year I was best up and comer, and then boom, everything imploded. B of A used to say, "Oh, we don't have any mortgage exposure." Then they bought Countrywide at the peak of the crisis.
By definition, is exposure.
Yeah. And then Merrill imploded. I went and traded derivatives. I traded volatility for a year and then was recruited to New Leaf Venture Partners. I was brought on to build their public trading effort by Philippe, who is one of the founders of the fund. And so from 2010 to 2014, I built and ran the public portfolio, one of the first venture funds to actually create a trading capability and run a public portfolio. I left in 2014, started Aleph Point Capital. It was a healthcare-based focused fund, trying to, at the time, use big data and algorithm technologies and natural language processing in healthcare investing. And funny, I was trying to raise capital, and at that point, people didn't actually think you could use big data. Today, it's obvious. Everyone has.
Honestly, I'm expecting solicitations in the chat like now.
Yeah.
Somebody's going to think that's a great idea and want to invest in that fund. Anybody that came on without realizing it was 7 years ago.
Yeah, 7 years ago, people thought I was crazy. But somewhat fortuitously, I came across this technology, became enthralled in it, really believed in it. And in late 2015, we launched what was then Incisys, today IN8Bio.
So how did you find the technology? So I want to kind of dip into that a little bit. So and we'll talk a little bit about what IN8 does, because so IN8, is it fair to classify it as kind of a next gen cell therapy platform?
Yeah, we are one of the leading companies focused on gamma delta T cells as a cell therapy platform. As well, we have a unique approach, what today we're calling our Deltex drug resistant immunotherapy, or Deltex DRI. It's a unique approach that I'll talk to you, but to answer your question, it was completely by accident. As I said, I had never an intention to become a biotech entrepreneur. Truth be told, at New Leaf, I had done much of the diligence in Kite and Juno in 2015 and 14 when they were still private. By 2014, I was convinced that CD19 and BCMA CAR were crowded. If you recall, that was the summer that Kite and Juno actually became public. If you actually looked in 2014, there was already 45 protocols filed with the NIH RAC targeting CD19.
Yeah. And everybody wanted to be like, it was either an armored CAR or something that would secrete a cytokine or something with an on-off switch, but it was all the same target, and everything just there was tons of them. It was everywhere, but nobody got off of the CD19 track. And I wonder if it was just because everyone was banking on kind of the clinical, the clinical outcomes, right? Because really, Juno and Kite just pushed and pushed and pushed further in clinical faster than anybody else did.
I think a lot of people have said, oh, why don't you use this validated target as a proof of concept? But that comes at risk. As we've seen with PASDR and ODAC, there's concerns about innovation. Nobody needs 65 PD-1 inhibitors. Nobody needs 50 CD19 inhibitors. And the way I found the technology was, I was actually, in 2012, Yamanaka won the Nobel Prize in Medicine for iPS cells. In 2014, if you can believe it, there was only 4 posters with iPS cells. 3 of them were from Fate Therapeutics, who today is probably a $6 or $7 billion company. But at that point, nobody cared. One of them was from a transplanter at the University of Alabama at Birmingham, who said, do you want iPS-derived T cells and NK cells? I got a guy who can make them. We spent an hour at the ASH conference talking over his poster, and we swapped cards. I didn't think too much about it. Ironically, 1 month later, in January of 2015, I was in my office during what was then Blizzard Juno. If you might recall, it was a massive blizzard, and it was the first time in 110 years that New York City shut down their subway system. I got a phone call and said, hey, remember me? That guy I told you about, he's in New York City. He's stuck. His flight got canceled. Would you like to meet him? And so I walked from my office which is at 28th and Madison, to the Waldorf Astoria, and took our who's our scientific founder, Dr. Lamb, out for lunch at the Lexington Brass. For 3 hours, we sat and talked about immunology, his view of the world, and his technology. These cells that honestly I had never heard of, these gamma delta T cells. and this DRI approach, and I became convinced.
Mhm.
And so that's how the company started.
That's fantastic. So what is it? What is a gamma delta T cell for the edification of our audience?
So the gamma delta T cell is a unique cell that bridges between the innate or early immune response and the adaptive or memory immune response.
Mhm.
And thus it has properties like both of them. As many people know, cell therapy 1.0 people were focused on the alpha beta T cells. They can generate memory responses, they kill effectively, but those cells are restricted by major histocompatibility complex, or MHC. So they don't kill anything unless they're programmed to kill through a matched antigen. On the other side, the NK cells are innate. They don't need prior programming. They can recognize cell stress ligands through NKG2D ligands and can kill directly without priming.
Mhm.
As well, because they're not MHC restricted, the NK cells can go from a donor to a patient without crossing graft versus host. Well, the gamma delta T cell is a little bit like both. It has a TCR, it's gamma delta TCR, but it doesn't require priming. Its binding domains are almost like antibodies, IgG. It has a vast array, I think 10 to the 17 potential combinations in its recombination of its TCR. In addition, it has an NKG2D. And so the advantage, we believe, is ultimately the gamma delta T cell, one, is potentially safer. And so because its normal role is to distinguish between healthy tissue and tumor tissue. The big challenge with organ cancers is that unlike the blood cancers where the tumors are freely floating in your bloodstream, in the organ cancers, they're intertwined in the organ.
Right.
With CD19 and BCMA is you wipe out the entire compartment. I don't select between healthy B cells and tumor B cells. I wipe out all of them in a process called aplasia. Unfortunately, if I have an organ cancer like brain, liver, lung, where the tumor is intertwined with the organ, if I wipe out the organ, I kill the patient.
Right.
And so the gamma delta T cell, we believe, can make that distinction. In addition, its receptors, it can target the heterogeneity of the solid tumors, and we believe they can be delivered from a donor to a patient without causing graft versus host disease. So a really unique cell set.
Yep, and it gets you around, in theory, this gets you around the need to have kind of multi-stage combinatorial therapy, right? If you start trying to if all you have is the CD19 T cell hammer, and you have to try and peel away the layers of everything else in order to make sure that they can actually get to the cells you want them to get to, you've now got, a 2-level, 3-level, 4-level combination, and that's a lot of switches to throw in the immune system.
Well, we think ultimately you're going to need a combination. And the interesting thing about cancer therapy is that if you look at how cancers have been treated, it's this progression of single monotherapies, right? I mean, we use this euphemism of this war on cancer, right? If you were actually fighting a battle, and unfortunately we're going through a lot in the news cycle today because of what's going on in Afghanistan and the Middle East. But if you're going to fight a battle, do you wipe out as many of the enemy as possible and then give them time to recover and rebuild their strength and then switch therapies? And so that's what we do in oncology therapy. But if you think about, as an investor, many of us have seen from other areas, if you go to the antiviral therapies, right? If you think about all the progress we've made in HIV and hepatitis, it wasn't until Gilead and others could combine therapies with Atripla or with Sovaldi, where we have multiple therapies with different mechanisms of action, each of them acting on the virus simultaneously, that we can actually knock it down.
Yeah.
Right.
And we believe ultimately it'll be something similar in oncology therapy.
Okay. Yep. That's really, really interesting. So where do you see, I mean, cell therapy, I would imagine, is still characterized as crowded, correct? So,
there are
how
dozens
do you,
of companies.
Yeah. So do you find it challenging to get people really kind of on board with the thesis, or get the thesis enough airtime where it's, understood, respected, digested, et cetera, where, then people can actually start talking about, results in clinical strategy rather than, trying to digest and parse what you're trying to do from a biology perspective?
I think it's really hard because you have this wave of people, many of them who are still on CD19, trying all different kinds of combinations of CD19, whether it's allogeneic CD19 with an NK cell or gene edited CD19 on an alpha beta T cell. There's a lot of people still stuck on CD19 and BCMA.
Yeah.
We took a very different approach. And I think it was very hard because we're the leaders in this space, and we've been bold and went out by ourselves, and people said, what are you doing? If this is so hot, how come nobody else is doing it?
Right.
Yeah.
Our approach, as I said, we call Deltex DRI, is an approach that allows us to combine simultaneously with chemotherapies, in particular alkylating agents. These are agents that function by driving DNA double-stranded breaks. And truth be told, when I first came across the technology, decided to start the company, the first few years, people said, you're crazy. Why are you going against chemo? Didn't you listen? Chemo's going the wayside. We're going to, targeted therapies. We're going to CAR Ts with SCFVs. And we're going to get away from this broad, nuclear bomb of chemotherapy. And truth be told, it's 5 years later, we're still using chemo. For 90 years, chemo has been the most effective thing because it works. It's that initial bomb. It can shrink the tumor. But what we found, what's interesting is when we first started, people thought chemo was going to go the wayside. But in 2017, Merck did something interesting. For the very first time, they got approval for a drug, Keytruda, across all indications at the same time. And that was in those patients who had mismatch repair deficiencies. And what they found is when they combined with chemo, whether it was non-small cell lung cancer, bladder cancer, triple negative breast cancer, chemo could elicit immunogenic cell death, and Keytruda was more effective across the board.
Right.
And this has been shown by Roche with Tecentriq and others with PD-1 inhibitors now. And that's what we find when we dose with the alkylating agent. The thing is, DNA double-stranded breaks are an evolutionarily conserved process in biology. Yeah. Our bodies are designed to eliminate cells that have DNA damage. And they can accomplish that either through simple DNA repair, through the triggering of apoptosis and cell death, but it turns out there's a backup. Simultaneously, those cells that have DNA damage upregulate the innate immune ligands. We know of at least 9 NKG2D ligands today, and we can upregulate multiple ligands simultaneously on the surface of those tumor cells to mark them for destruction by the immune system.
Yeah, so it's I listen to you talk about this, and I think about the knowledge progression over the past 7, 8, 9, 10 years in terms of T cell biology, right? And I remember vividly when people would talk about Opdivo and Keytruda, the analogy of like taking the brakes off of the immune system, and they function on CD8 T cells. And as we went along, we realized, well, they actually hit a lot more than that. And oh, by the way, there's a bunch of other kind of T cells rather than just CD8 and CD4 T cells, right? There was a brief period where they were characterized as like innate lymphoid cells. It's like catch-all thing. It wasn't CD8, it wasn't CD4, and it wasn't CD17. It was something else. And now I think we've probably grown quite a bit where the same people that told you that chemo is going away, we've got this other stuff, they'll probably come back and have some other obscure biology thing to mansplain to you that you're doing it wrong.
Look, I think the thing about biology is it's so complex, and what we have to realize is we don't really understand it. Yeah. What we actually know is probably like this. I love this approach because when we're thinking about targeted therapies, eventually the tumors become resistant. And part of that is because I always liked it when you go to scientific conference. If anybody remembers the Roche biochemical pathways chart, it's kind of like the map of the Tokyo subway system. It doesn't matter if you take down one line or 2 lines. There's always a way around.
So how did you all decide to start with the 2 targets that you have with glioblastoma and the particular leukemia?
Biology and the FDA. To be frank, the holy grail that everybody's trying to get towards is to get towards allogeneic or off-the-shelf cell therapies for solid tumor cancers. There's a lot of complexity in that to get there. And truth be told, our scientific founder, Dr. Lam, initially went to the FDA and said, oh, I'd like to run a clinical trial in solid tumors in the upfront setting with an allogeneic product that's genetically altered in the brain. And the FDA was like, well, hold on, let's take a step back. Let's manage this reasonably. And so on one side, our INB-100 program in leukemia is not genetically modified, but it's the first gamma delta T cell program to deliver such a high concentration of donor-derived gamma delta T cells. Right. So really trying to test the safety of donor-derived cells. Right. On the other side, INB-200, our solid tumor program, is in newly diagnosed glioblastoma. It's autologous, but it is also genetically modified. It's the first genetically modified gamma delta T cell program to enter the clinic. And so we can test the two separately, and then ultimately we'll try to bring them together. Why did we go into GBM? Look, I know a lot of investors hate GBM. It's kind of like Alzheimer's and or ALS. Very difficult indication, but part of that was driven by biology. When we ultimately studied the biology of intravenously infused T cells, what we found is there's a hub and spoke model of T cell trafficking. When I infuse a bunch of T cells into my arm, they actually traffic to the hub, which turns out to be our lungs. Mm-hmm. It goes to the lungs, and then over time leaks out to the liver and the spleen, and then may traffic elsewhere. But as we all know, when we look at cell therapy, everyone does this assay where they show an effector to target ratio, or E to T ratio. Mm-hmm. And we see that 1 doesn't do much, 5 doesn't do much, 10 doesn't do much. But when you start to get to 20, 40 or higher, you see really more robust responses. If 95% of my T cells are stuck in my liver, in my lungs, I'm not going to get to that high ratio, especially if I'm trying to treat something like ovarian cancer or pancreatic cancer. Right. By going into the brain, I can bypass the trafficking question and put the cells directly into my tumor bed. It's one less variable in my hypothesis test for my clinical trials. Okay. So we know that cell therapy can target tumors like glioblastoma. The problem is tumor heterogeneity. They can't cover the totality of the tumor. Right. Our particular approach with this DRI, we know we can upregulate multiple markers on the surface of the tumor. And so we eliminate, by going into the brain, we can eliminate the question of trafficking and delivery, which is major. Right.
So I want to kind of wind back a little bit to, when people were telling you were crazy, right? And not so much just from a personal standpoint, although we can get to that too, but talk a little bit about basically kind of trying to raise the money in chunks when, I don't know what fraction of investors or other people kind of, told you either you were crazy, this idea is not going to work, your biology is wrong, your strategy is wrong, you haven't done this before, we just don't like you, I've never heard of you, like all of those things. Like how is it to basically try to get this stuff done when your biological thesis is solid, you have a lot of people telling you it is solid, you have a great bunch of people behind you, but to just have people tell you that you're wrong and it's not going to work?
It's hard, and I've heard it all. We probably went to 200 VCs, maybe more, and we've heard no, oh, we'd like to invest but we need more data. It's absolutely incorrect. What's humbling is, and what's helped is I was an investor early on, right? Yeah. I had a 16, 17-year career on Wall Street before launching this company. And as an investor, I've had multiple occasions where I sat there in an investment that was, to be frank, really lonely. Nobody believed in it, right? When we first looked at Fate in 2014, nobody believed in it. In September of 2016, I convinced the analyst at that point at Franklin Templeton to do a PIPE in Fate at $1.96. Last year at some point, I think that stock hit $110. But I can tell you, back then, Nobody cared about it. A local New Jersey company a few years ago, Celator, in 2016, they were trading at something like $1.12. Nobody believed in it. 4 months later, after phase 3 data, they got sold for $30.25. So I was at least experienced enough to understand that often people don't believe in it. In fact, as an investor, where I made the most money, it was when nobody was around. Nobody believed in it. So you can find an asset, and when everybody was coming in your direction, you knew you were doing the right thing. There's an old joke I always kind of laugh about is, how does science progress? And the joke goes, one funeral at a time. Because basically in science, someone comes up with some research that becomes dogma. And during that person's lifetime, that's what's the belief in science. And then that person passes, someone else takes the torch or the baton, does more research and figures out, oh, something about that was actually a little bit off. You know, Jim Allison went around pitching the same handful of companies or patients for, almost 30 years. Nobody believed
decades.
It. And then, oh, he has a Nobel Prize. Judah Folkman went around talking about VEGF. Nobody believed him. And then, oh, we have Avastin. And so you have to be patient and persistent.
Yeah, the graph from this is a terrible idea to of course, takes, and it's the stuff that everybody believes, right? To paraphrase your asset trick, your trick is to find an asset that everybody believes in before they believed in it,
Problem is, then you invest in the 60th PD-1 inhibitor.
Right.
I guarantee you anything from 8 to 60 are probably all going to lose money.
Yeah. Well, it's funny. I remember when a couple of startups ago, I think we had like the 8th or the 9th one or something like that, and strategizing on what to do with it. And you saw this wave coming. Everybody was developing one, and we had the idea of, well, and we were too early to implement this idea, sadly, but I think it would have worked. We basically said, listen, let's just basically partner ours and more or less give it away, because people are just going to fight over what to combine it with. And ultimately, if you look at the data on PD-1s, they mostly all seem to work just about the same. There doesn't seem to be not a lot of difference. So, but anyway, back to your stuff here. So it's interesting that you bring up the one funeral at a time thing, because the scientific community does not go against its leaders terribly easily when they're around, right? Because they will either they'll call up the head person at Science or Nature and tell them that the particular paper that they're thinking about is wrong or whatever it is. It is very, very difficult for a counter idea, even if it's correct and even if it has a ton of data behind it, to get any traction.
Yeah, agreed.
And it scales, right? It's not just the VEGF stuff, but even, I imagine you see smaller flavors of it with, why would you do anything other than CD19? Because nothing else has, demonstrated clinical efficacy yet. So why would you do anything different? You should do CD19 as a pilot one to show everyone that you can do it.
To your point, I know there's probably some scientists out there thinking about their technology and whether they can become an entrepreneur. The important thing about science is science doesn't care what color you are. You can be black or yellow or white. It doesn't matter. All science ultimately cares about is this right? It's harsh. That's all science cares about. And if you believe in the level of research that you do, that the depth of your knowledge is deeper than the next person, then the important thing is persistence, right? Really going through this whole process. If you've read Steve Jobs' autobiography and some of the things that he said, the difference between people who are successful and not is persistence. They just refuse to give up. And I think that's a big part of becoming an entrepreneur and starting up a company.
Yeah. So if you go back to the point where you actually, decided to do this, right? What was your, because you were the head of a venture fund, right? I mean, how do you turn around and say, this is the thing that I have to do? And then I don't know how many other people were at the fund or anything like that. How did you think about that transition? How did you think about what you wanted to do? And when you did, how did you make that transition? Because that's it's one thing as an investor to say, the typical path is to say, this is a great idea, we should start this company. I will write a check and then we'll put a management team around it, right? But how did you obviously did that differently. What was your thought process like?
It wasn't my thought process. It was honestly a mentor. So interestingly, I read one of my largest investors. I read an article recently on Medium, and it was about how do you become successful? How do millionaires become millionaires, and how do billionaires become billionaires? Nobody's born with the knowledge of how do I become successful. People are taught that, and the easiest way to be taught that is to find someone who's done it to give you advice about their process. And the same thing goes from millionaire to billionaires. Find some mentors who've done it so that they can go through and push you. And really, is one of my investors at the time in the fund sat me down and said, "You're going to fail. You are doing too much. Things are crazy." It was 2016, if you recall, January of 2016, the XBI was probably down about 50%. I was struggling to find investments and keep performance up, and trying to launch this company. And at some point in the middle of 2016, actually my performance was actually really strong in 2016. It was actually positive when the biotech indexes were down. But my mentor said, "You're going to fail. Pick something that you truly believe in. Which do you truly believe in? Which do you think you can make a difference? And focus on that, because you're spread too thin."
Yeah.
And so I sometimes I see people who say, "Oh, we've encountered people. I want to be an entrepreneur, but I want to keep my tenured professorship." It's too hard.
Right.
To straddle both sides, you're not focused, and it is so difficult. You have to pick the one in which you believe in, which you will be happy with, and which you think you can make a meaningful difference, and go with that one.
Yep.
So I didn't consciously make the decision. I was actually told, "You're going to fail," and that was enlightening.
Yeah.
Well, it made you evaluate things differently, right?
Yeah.
It wasn't about just keeping pushing and pushing. It made you stop and think, okay, what is my highest and best use in this scenario?
Right. And it's funny, right? Because people, I think, get caught up in a lot of the romantic notions behind what they're doing, right? And making the choice of what not to do is actually just as hard as making the choice of what to do, because it's really easy to sell someone on an idea, right? There's all sorts of positive stuff. You could talk about the potential, you talk about all of these things, but actually selling them on the execution is much harder, because like you say, it's like, well, that means you cut out all this other stuff, right? So so no more, you can't be a professor anymore, you can't do this, you can't do that. And it's funny, there are very few examples of people that have made that work. So if you were to go back and do it again, I mean, what were some of the hardest things for you in making that transition?
I think the whole transition is hard. Even today, it's kind of like it looks smooth on top. It's like the duck, right? It looks smooth on top, but underwater, you're battling every single day. For us, I take the mental viewpoint that if it can break, it's going to break. The question isn't if it's going to break, it's when, and how do you plan for that? And so I think the whole process has been difficult. Launching a company, being a first-time CEO, there were times when we were, out of money or near not making payroll. And there were times when biology just didn't work. The initial CMC for scale up, one of our advantages, Dr. Lamb has been in cell therapy for 30 years. He trained as the first postdoc under Adrian G., who's considered the godfather of cellular therapy manufacturing. And with that, we finally got through the manufacturing processes and the scale up, but there was a lot of failures, a lot of head banging on the wall and furniture thrown by Dr. Lamb, but we got through it.
How did you, so you were in New York, he's in Birmingham, right? So how did you decide the sort of the nuts and bolts process of building a company and what you would put where and how to do that? Because obviously New York, we are biased in favor of growing things here in New York, right?
I think I was always in New York. I've been here for over 20 years. And so I wanted to stay in New York. My family's in New York. My kids are here. Birmingham, Alabama was where our chief scientific officer, Dr. Lamb, was located. At that point, he was a professor of medicine, had his own laboratory, and built and ran the GMP facility there. For us, it just made sense to keep our R&D down there. We've continued to expand R&D down in Birmingham. It's been a little bit cheaper for us to, both lease and build out lab space. Yeah. But at the same time, we've built our business operations here. We're actually going into a new facility, a new space in the Empire State Building soon. And so we are expanding here. And ultimately, we will build additional facilities here. There is a ton of talent here. There's major universities, Rockefeller, Sloan Kettering, Columbia, NYU, with research and among others with research capabilities. And so we will ultimately build labs here as well.
All right. Well, that's very good to hear, and the folks from the EDC will both thank us and make sure that you do so. So winding back, if you think about where you are now. So actually, before we get to that, so you're now a public company.
We're now a public company.
Yes. Yeah. So we've talked about the trials and tribulations of getting the company started and off the ground, but I'm actually really interested to hear about the process of becoming public, especially in New York, right? Because we will stand on the soapbox and shout this for ages, but we've long believed that the proximity of companies to the investment pool in New York, certainly from a public market standpoint, is a huge advantage. So can you talk a little bit about just about the process of going public, and really from where your company sits, right? So how did you get this story of biology that is outside the mainstream acceptance into the minds of investors enough to basically complete an IPO?
It was tremendously difficult. Look, I do have the advantage of having been on the other side, knowing a lot of the investors. I think the advantage of being in New York is that you are close to many investors, in particular public ones. But it's tremendously difficult today, especially to do what we did, which is to launch an IPO without a crossover round. Mm-hmm. And it was purposeful the way that I ultimately funded the company. We didn't get seeded by big VCs. We never did a big splashy round where you see they raised 50 million in a series A, and then it goes to like 150 million in a B, and then subsequently 150 million IPO. We were measured in our capital raising. To date, we've only raised approximately 77 million in totality, but we were also very efficient with our capital. The big challenge is ultimately, there are so many deals coming out today that a lot of people are taking the shortcut and they're looking at who the current investors are. And so ours was much more difficult because we didn't have those investors, and partially because there's always a balance between, look, if you raise 500 million and you're sitting there in preclinical or just discovery stage, really the only asset you really have is cash, right?
Yeah.
And unfortunately at that point, then the equity is all held by those who gave you the cash. As someone who's been in the business for 20 years, that wasn't exactly where I wanted to be. I wanted myself and my management team and all of us, my board members included, to hold more of the equity of the company ultimately.
Yeah, I mean, that's quite honestly, that discussion is an hour in and of itself, right? Because it is, well, because it's exceedingly rare, and you have funds that are going to put more and more capital in, and surprisingly they want more equity for all of that capital. right? So I mean, I think about the company that Flagship launched, the circular mRNA company. Just they had a $450 million Series B round yesterday. So it's unclear to me that management still owns 20 to 40% of that company. I don't know that they do. So it's
Probably
very
not.
hard. Probably not, right? So it's very hard to, because if you don't want to do that, right? If those are the deals that are available and you don't want to do that, you've now limited you've limited your investor set, right?
Yes.
So, and people don't think about this that much, where if you don't have the splashy Series A, you kind of miss out on the Series B investors that invest in splashy Series A companies, right? So the subset of investors that you go to are different depending on the actual path that you took to get there in the first place, right? So that in and of itself is hard, because also the general prevailing sentiment is that there's cash everywhere. You're having a hard time raising capital. Why? Everyone is raising capital. Look around.
Look, we went to some of the well-known VCs, and one name in company formation that we knew, they said, we want 10 to 15 million to get proof of concept and get these INDs, and said, we're not interested in 10 to 15 million. Our funds are bigger.
Right.
We have to do at least 50 million, and we like to do it at 5 million pre. And we like to put our own management teams in.
Yeah.
Well, the problem I have is this process is so difficult. If the management team doesn't have skin in the game, it's too easy for them to just get up and walk out to the next company.
Right. You've been doing this for 5 years, so that sounds great. Why don't you crush the value of my company down to 5 million, and I'll move away for somebody else. You're welcome, type of thing. So doing it differently is much harder, because again, you go against public perception, and there's people that regard, and that isn't even the denial isn't based on anything you're doing as a company, right? They could tell you're doing the exact same thing. They could absolutely love your biological thesis, but just from an investment structure perspective, it doesn't fit.
Yeah, and that's a lot of the cases. Like many of the VCs today have gone into company formation, and for many people that works. And for truth be told, it's probably easier. You can spend your time focused on the science and the development of the technology, and not worried about trying to finance and the worries about not being able to make payroll or whatnot. It took a lot of work to get through it. IN8Bio today is through that. We're in a much more comfortable place. We have the capital to get through the milestones in the next 2 years or so that will drive value inflection points. But it was not easy.
Yeah. So if you had somebody come to you tomorrow who's they're working on either gene therapy, cell therapy, CRISPR stuff, they're doing this, they're a postdoc at Mount Sinai, and they say, I want to go start my own company. So what do you say to that person?
So I have numerous people who've come from Wall Street or other places that I've known for years who have come to me, and they've asked, how'd you do it? Right? It went from starting a fund to starting a company is the hot thing to do, to be a founder. And a few of these people I've said, the first thing you should do, the best advice I can give you is go home and talk to your spouse. Are they really in this with you? Because it can be so difficult. In the worst days, the company was almost broken into pieces. My wife was at home lying on the couch with the migraine due to the stress of everything that was happening. We did it even more difficult. As we launched the company, we had our first child. And then as we were going through a private round, we had our second child. We had daycare, the company burning cash. At some point, I was funding the company out of pocket. And so I don't recommend all of this, but really having a spouse who's in there. I came from the Wall Street world. And if you have, lifestyle expectations of up here going through this entrepreneur process, things have to get tightened down.
Yeah.
And if your spouse isn't behind you on that, you really need to have the support, because it's too difficult, I think.
I would 1,000% agree with you. Having had not the exact same experience, but similar ones, almost down to the timing of children, right? And that is non-trivial, because you're going through some stuff. You're going through stuff with all of that anyway, and like we said, to layer on top all of the challenges that you're going through with here, I don't know which part of it is too much, but taken all together, it becomes too much. It becomes really, really difficult.
That's like
That being said, there's never a good time. There's always a bad time.
Yeah,
you always kick the can down the road. If you really want to do it, you may as well do it. And even if you fail, you always learn.
Yeah. With both of my kids, I took a little bit of vacation, but no maternity leave. And it's and I'm always like, do as I say, not as I do. I learned the hard way. How to things you should do and things you should not do. Go ahead.
No, you go.
Oh, yeah. So I was going to say, so as you obviously, so she bought in. It wasn't easy, but right, she bought in.
She bought in.
And you've come out the other side. Talk to us a little bit about building out your teams, right? Because that's a frequent conversation that we have with our guests, particularly our entrepreneurial ones. How did you go about that?
Building out a team is difficult. And look, we've made mistakes early on. Early on, especially when you're an entrepreneur, you have no money, you don't have any resources. So you tend to take what you can get, right? And we've learned through some of those processes. Look, not everybody is equal in their skill set. I'm fortunate in that I've had a broad range of experiences. So I have relationships along Wall Street. I can do financial modeling. And Dr. Lamb, my scientific founder, had regulatory and manufacturing and other experience. So we could synergize. We've made mistakes. I think the important thing early on is to hire people who don't just want to be a manager, but can do things. Right. In many companies, a small portion of the employment, the employees are the ones who are there to do things, who don't care whether there are challenges. If you told them to go through that wall, they're going to keep banging their head on the wall till they get through it, and they will solve those challenges. I think early on in an entrepreneurial environment, you don't have the luxury to have a lot of people around who don't do things or can't do things. We've had hires where they want to be hired and they look great on paper, but they needed an additional team. Yeah, that can work later, but early on, you need people who can have the knowledge and who can do things.
Yes. If the first thing they're going to do after you hire them is hire 3 more people to be part of their team, it's perhaps not exactly the capital efficiency that you're looking for that kind of output.
Yeah. And you have to realize you have to early on. I think you have different team members for different
phases of the company.
And phases of the company. And early on, it's people who can exercise, who can execute, who don't mind doing a little bit of everything, no matter what their titles are or what their roles are. I mean, hell, during COVID, I was walking around our office wiping things down with the sanitizer, right? Because we all have to pitch in to make sure that the company can move forward.
Yes.
Right. So making sure people are tolerant and understanding of the risk. I always ask a question when we hire people: what are your spouse's concern? What's your family's concern? Because it's not just an individual's decision, right? Because it impacts their family. If I fail, they fail, and it impacts their spouse and their kid.
Yeah. That's really interesting. I don't know that I've heard of anybody that's kind of taken that approach before, but it's a really smart one. Because the last thing you want is somebody who seems to be all in, on paper and when they're there, but for you to realize that, they actually haven't mentioned to their wife that they're going to go join a startup. It hasn't really been a topic of the conversation, right?
We did the opposite once. My husband left DC, moved to another city, took a job, and we didn't know if he'd like it, and so we didn't tell anyone at Glaxo at the time where I was that he was going. So we flipped that scenario.
I was scared for a second there. I thought you were going to say he didn't tell you. I was going to be like, oh gosh.
Then he would have been my ex-husband.
Yeah, no, I was very surprised.
Look, we ask questions, and sometimes difficult questions in the recruiting process, because some people look fantastic on paper, but there are issues that just, maybe it's just at this point don't make them a fit, and that's okay.
Yeah,
It's okay to lose people, because it's better to find out now rather than later.
Yeah, absolutely.
Yeah. You know, I think also there's a lot of romanticism caught up in either starting a startup or joining a startup, where people want to tell you the positive things. They want to talk about how much they love the mission. Like there's a lot of positivity wrapped up in that. And if you don't actually get to the hard questions there and then, you know, the hard stuff is going to come, but you better ask beforehand, or though you kind of won't, you won't know what you have until something hits the fan.
Yeah.
Yeah.
I mean, being an entrepreneur and in a startup is sexy, but you know, it's difficult. It takes a huge commitment on your life, and it's hugely stressful, right? Look, I think recently Elon Musk, talking about Tesla, said it sucks. The worst points of my life were the, you know, parts of the manufacturing expansion where he was living there 24/7.
Yeah. But as we plug in our Model Y, we're going to get it, right?
Yeah, no, you know, I don't feel that bad for Elon Musk, because he could leave tomorrow and not be the worst for anything. So his daycare his daycare bills are not a problem, right? So, all right. So to transition a little bit here, we think and talk a lot about, you know, kind of the impact of what we do on patients, right? And you're now working in areas that, you know, where, you know, this is not a standard solution for the patients that you're talking about. So can you tell us a little bit about, you know, how you've approached either your company's connection with patients, or at least kind of the broader medical community, of how you're actually getting these therapies to the people that need them most?
So today in our brain tumor study, we're partnered with Dr. Bert Neighbors. He's the head of neuro-oncology at the University of Alabama in Birmingham.
Mm-hmm.
What's unique is our particular approach is added on or layered on top of the standard of care. So the patients are in the frontline setting, they're newly diagnosed patients, and they get what's known as the Stoop regimen. The standard of care in glioblastoma has been the same since 2006, when it was first published in the New England Journal of Medicine. So 15 years, it has not changed. Unfortunately, we know that in glioblastoma patients, all of the patients will ultimately die of their disease. The 5-year survival rate is in the single digits, about 5%, right? And most of them relapse within centimeters of the original resection cavity. And so we know standard of care will shrink the tumor, but there's always microscopic residual disease that will grow back and will ultimately kill the patient. Our approach is layered on top, and the advantage is, when you've finished treating with standard of care, or as you're going through standard of care, reality is anything that's sensitive to standard of care, you've already killed. Whether it's through surgical resection, through chemotherapy, or radiation, anything that was sensitive is already dead. What's left is pretty resistant to the standard of care. What's left are chemotherapy resistant cells and the so-called cancer stem cells that might be quiescent. What's interesting about our approach is, by layering on top, we know we can actually upregulate these immune markers on the surface of the tumor, even if they're resistant to chemotherapy, and on the cancer stem cells.
Right.
The goal is to drive to a deeper response. Because if 90% of the tumor can be removed through surgical resection, an additional 90% removed through chemotherapy, it's that 1% that grows back that kills the patient. If you can drive to an even deeper response or eliminate it, like we saw in our animal models, we saw complete eradication. It's getting that residual 1%.
Yeah, it's driving.
And trying to target it when it's at its weakest, not letting that 1% grow back to 100%, and then trying to target it, because that's much more difficult.
No, this goes back to your analogy from before, where you want to turn that 1% into 0.1, 0.01, rather than wait until that 1% comes back to 10% or whatever, to then try and go around and do it again. So, I mean, this is a really difficult patient set, right? This is a group of patients that, frankly, doesn't have a lot of hope and they're dealing with they deal with a lot here. So how does, do you interact directly with those groups? Do you get outreach from folks? How does that stuff sit with you?
We get outreach, and it's to be frank, one of the most rewarding parts of going from an investor to an entrepreneur. I never realized it until I started getting emails from patients or their families and how meaningful it is. When you're an investor, if you're right in the phase 3 trial, it's like a flip of the card. Oh, I made a great bet. Yeah. Here, right. You actually have people emailing me. My wife, my child, my parent has glioblastoma or AML. Can you help? How can we get them into a trial? Can we? Yep. And that's much more meaningful. For us, it's trying to target those residual tumors. Of our phase 1 trial, of the first cohort that we dosed, all 3 of the patients went months beyond their expected median progression-free survival. And so that's encouraging. Our first patient that was treated, he went out over 15.6 months before he unfortunately died. Unusually, he didn't die of a brain tumor. How often can you say a glioblastoma patient didn't die of a brain tumor? He had a pancreatic cyst that resulted in an infection and died from that, but completely separate from the brain tumor. And so if we can make a difference, we just keep pushing forward little by little. We know in glioblastoma, small improvements in progression-free survival or overall survival can get you to an approvable product. Early on, I had a discussion with a CBER reviewer, and they said, look, it's to the FDA, it's about benefit risk. And we know what happens to these patients if we don't do anything.
Yeah. Right.
I was just going to say, so, and those of you who knew her, Dr. Barbara Murphy from Mount Sinai was a personal friend of mine, my neighbor, but also was a friend of NewYorkBIO. And we actually had her on our webinar last summer, and she died this summer from glioblastoma. And so it hits home in lots of different ways, but I know the patients are appreciative of the outreach you're doing and trying to understand their journey as part of your preparing treatments and cures.
Yep. It's hard. I don't believe in avoiding something just because it's hard, because we have to try to make a difference. If people aren't willing to try to make a difference, we'll never make progress, right? Yeah. If you watch the Olympics, how do the high jumpers? They don't jump over that bar the first time. They fail and they fail and they fail until they get to where they are today. We'll keep trying as long as the science makes sense, the biology makes sense, the pathways and the data continues to support those pathways. We'll keep trying because we believe we can find the path to actually making a difference.
Yep. Well, listen, that's fantastic to hear. And, there are, we see this all the time. We think about different pockets of patients, and, the path that you're blazing is a hard one, and there's probably hard stuff to come. But like you said, science is done in a stepwise manner, and if you don't fight through all of this, there's no hope for them getting it at the end of it. So I'll leave it on that. One of your Curagen colleagues, Cindy, said hello, and it's great to see you. And I just want to say, as somebody who's I've seen you present this company numerous times, and you mentioned the duck analogy before, where everything looks calm on top of the water and you're working really hard under it. I know things have been really difficult, but quite honestly, like, you make a lot of this stuff look easy. You make the thesis easy to digest. You see what the benefit is a lot, and frankly, it's really easy to root for you and what you're doing. So from all of us, congratulations on getting where you are now, and we're all pulling for you to do well the rest of the way.
Thank you.
Thanks, Will.
Stay tuned.
Thanks so much.
Yes,
thanks, everybody. Have a great day.
Bye, everyone.
Bye.
Thank you for tuning in to NewYorkBIO's virtual breakfast series. Join us every Tuesday at 9 a.m. for more discussions with leaders from across the healthcare spectrum. For more information on NewYorkBIO, please visit us at www.newyorkbio.org.